Welcome our viewers to the first part of Form 4 genetics. We shall be dealing with this genetics in two parts, the part 1 and then part 2. I am your teacher, Mendel. I'm a teacher of maths and biology, and therefore feel free. Let us move together to the end. So in our first part of genetics, that is part 1, we are going to start by looking at the definition of this term genetics. Now, genetics is a science which deals with the study and understanding of heredity, evolution, development, ecology, molecular biology, and forensic science. That is the broader meaning of genetics. But in simple perspective, genetics is simply the study of inheritance and variation. The study of inheritance and variation. Now, there's this man by the name Gregor Johann Mendel. This man is believed to be the founder of genetics because after performing a lot of experiments that we are going to look at, he was able to prove that actually some of these characteristics are inherited and how they are transferred and how some of the offspring were very different from the parent, thus variation. He first demonstrated the inheritance of traits in pea plants. So we are going to look at a lot of pea plants, and later it was referred to as the Mendelian inheritance. So after he did all those experiments, it was therefore moved, and as per now we call it the Mendelian inheritance. The main concept behind studying genetics, why do we study genetics, and what are some of the concepts that I want to learn through this genetics, is one, it explains how the traits are passed from parents to their offspring. We shall be able to know how some of the characteristics we possess were passed from parents to us, how some of the characteristics were not passed, and how maybe we are different from the others. It also explains about the gene and the number of chromosomes present in an individual with their importance. We shall look at the chromosome number and in an individual, and perhaps what is the importance of those chromosomes. Now, there's also another term that is variation of species. Variation of species. Now, the term variation can be defined as the range of differences between individual organisms. That is, I can repeat, the term variation can be defined as the range of differences between individual organisms. How you vary, how one organism varies from another organism. Now, this variation occurs within species. For example, the difference between individual human beings. You may discover that even if we have identical twins, you may find that they have some features which they may not be sharing. That's what we call the individual differences in human beings. Brothers and sisters from the same father, from the same mother, having difference maybe in terms of height, body size, skin color, etc. We're going to look at those. So those are what we call variation within the species. There are two types of variation. One is known as continuous variation. Now, continuous variation, in this variation, all types of features have a normal distribution. All types of features have a normal distribution. For example, the height of individual human beings, width of a tree, weight of a cat. Example is that one, that is the height in individual, and therefore also we have the leaf sizes and the shapes. They are continuous. Now in discontinuous variation, there are only a few features which fall under the category of discontinuous variation. For example, the different color of flowers in a single plant species, different types of blood groups in an individual human being, different types of earlobes, etc. When you're seeing there are very different types of earlobes, those are called discontinuous variation. Discontinuous variation. Also we have the flowers, the same plant but with different types of coloring of flowers, so that is discontinuous variation. Now let us talk about the chromosome. Now what are these chromosomes? We want to understand what are chromosomes. Now generally chromosomes are thread-like structures which are found within a nucleus of a cell. They are dark and contains an organism, that is organism's genetic materials known as DNA. Now most of the time the chromosomes are too thin to be seen except with an electron microscope. But when a cell is dividing, they get shorter and fatter, so they can be easily seen with a light microscope. And that's why when we are preparing our slides in the lab, we tend to take the apex of the growing shoot or the growing root, and then we prepare slides from that and we observe under light microscope, and we are able to see some of these chromosomes. Why? It's because they have become shorter and fatter. Now let us talk about the chromosomes in our nucleus as a human being. Now the chromosomes in our nucleus are packaged in the form of DNA. So when we talk about chromosome, these are just packaged form of DNA in our nucleus. Therefore, the DNA normally exists in what we call a non-condensed form in the cell's nucleus. We say it's always, we use the word apparite. Now, it condenses into chromosome at the center and lower left during cell replication. And also you need to know that human cells contain 46 chromosomes. Now which are in pairs, so they are in pairs, and the sex cells, what we call this, that is the sperm and the ova, contain only 23 chromosomes each. So the 23 chromosomes comprise one from each pair. That's why we're having the 23 chromosomes. It comprises one from each pair. Now, of the 23 pairs of chromosomes present in each human cell, one is the sex chromosome. So we are saying the out of 46, each one of them contains a pair of the 23 chromosomes, which forms the egg, and which forms the ova and the sperm. Now, this determines the sex of the individual. Male have what we call the XY. The shape looks like XY. Then the female have XX, capital X, capital X. So the presence of a Y chromosome results in a male features developing. So that's why we have the Y chromosome representing the male, because of the presence of that Y chromosome. I hope you have understood that one clearly. So that is a chromosome. Now, when we move on, what about our DNA? We also talked about a chromosome. What is now a DNA? DNA is often called the blueprint of life. And in a simple terms, DNA contains instructions for making proteins within the cell. So that is how our DNA looks like. And just maybe to elaborate more on the DNA, that each chromosome contains one very long molecule of DNA. Each chromosome contains one very long molecule of DNA. The DNA is an abbreviation for what we call the deoxyribonucleic acid. Deoxyribonucleic acid. Now, the DNA molecule carries a code that instructs the cell about which kind of protein it should make. And each chromosome carries instruction for making many different proteins. Okay? So from the DNA, because the DNA carries that code that instructs the cell about which kind of proteins it should make, so therefore each chromosome carries instruction for making many different proteins. Because remember, it is the DNA which is found, which are located on a chromosome. So one chromosome will be carrying a lot of instruction for that. So that is, and when you look at what we have here, this is a photograph of a DNA. That's an example of a DNA. Now, what about a gene? We have talked about chromosome, and what about a gene? So what is a gene? Now, a gene is that portion of a DNA which codes for a protein. Now, chromosomes are made up of several genes which occur in pairs. The total set of genes in a cell is called the genome. So we'll be hearing about the genome. That is the total set of genes in a cell. And that is typically how a gene looks like. It has been expanded from the nucleus. So you can see the cell, then from the nucleus we have the chromosome, from the chromosome we have now the genes, and therefore this chromosome carries, the gene carries all the instruction to form, instruction together with the chromosome to form a respective protein. So next I want us to look at the differences between a DNA and RNA. Now, a DNA has what we call a deoxyribose sugar, and RNA has a ribose sugar. DNA bases are A, T, G, and C. A stands for adenine, T for thymine, G for guanine, and C for cytosine. While the RNA bases are A, that is for adenine, U is for uracil, G is guanine, and C is for cytosine. Now, the DNA are double-stranded, while RNA is single-stranded. DNA is found in nucleus, but RNA is found in both nucleus and cytoplasm. DNA is stable, while RNA is less stable. Now let us continue and look at the definition of some of the terms we are going to use here and be able to understand them. The first term is an allele. What is an allele? An allele is an alternative form of a gene. Pairs of allele occupy the same relative position on chromosome pairs. Gene, I've already talked about it, is a section of DNA which codes for the information of a protein controlling a specific characteristic of the organism. Haploid nucleus, that is a nucleus containing a single set of unpaired chromosomes, e.g. in sperm and ova. In humans, the haploid number is 23. Haploid from the word half. Haploid, half. Diploid, that is a nucleus containing pairs of chromosomes. For example, in somatic, what we call the body cells in humans, the diploid number is 46. That is, our somatic cells have 46 chromosomes. Then you have another term known as genotype. Genotype is basically the genetic makeup of an organism. E.g., we talk about tall T, that is T capital T small, where T and T are alleles of a gene. Then we have the phenotype. Phenotype is generally the characteristics visible in an organism, controlled by the genotype, e.g. a tall plant or a dwarf plant. Then you have homozygous, that is having a pair of identical alleles controlling the same characteristics, e.g. double T, capital, where T is for tall. The organism will be pure breeding for that characteristic. Then heterozygous, having a pair of dissimilar alleles for characteristic, e.g. capital T, small t. Dominant, we shall use dominant too. That's a gene, for example the capital T, that always shows up in the phenotype of an organism, whether the organism is heterozygous or homozygous. But a recessive, the last one, is a gene, e.g. the small t, that only has an effect on the phenotype when the organism is in homozygous state. And now I want to move on, and I want us to look at the first law of heredity. Now, this is also known as the law of segregation. Now, this law states that an organism passes genes from parent to gamete with equal frequency. It states that organisms passes genes from parents to gamete with equal frequency. Now let us look at what Mendel experiments in what we call monohybrid inheritance he was able to do. Mendel, as I said earlier, worked with peas, and here's some reason why he did that. One is because they were available in many varieties. Also, when he used these peas, they gave Mendel a strict control. He was able to control over which plants mated. And also, fortunately for him, the pea traits are distinct and were clearly contrasting. So you can see there, that is what he used to do. He was trying to removing and replacing the male and female parts to make sure that there was no self-fertilization. Like from the first, number one, they are saying the removing stamens from a purple flower so that it cannot self-pollinate, so that the white flower can be able to pollinate the purple flower for him to get the results, expected results. Also, to test the particulate hypothesis, Mendel crossed two breeding plants that had two distinct and contrasting traits. For example, he used purple and white. So my question is, what is meant by true breeding? So when he was crossing, like for example in parent, that is parent plant 1 and parent plant 2, you can see the assumption was this is pure breed. That is, the purple was crossed with this white. And then the result was, that is when Mendel crossed fertilized his plants by hand. One of the questions he asked is, why is it important to control which plants will serve as the parent? So when he did that, the result that came out, he was able to find out that if he crossed from the true breeding pair, that is a purple with a white, the first generation, we are calling them F1 generation, that is hybrids, all plants had purple flowers. Then what Mendel did, he went ahead and self-fertilized the first one, that is the offspring of the first generation. When he did that, he obtained a second generation. And the second generation, he found that there was a mixture of purple and white flower. But there was a ratio, that is 3:1. A ratio. Out of every 4, there was 1 which was a white, but 3 of them were purple. So, he discovered for each monohybrid cross, Mendel cross, fertilized through breeding plants that were different in just one character. In this case, flower color. He then allowed the hybrids, that is the F1 generation, to self-fertilize. Now, in a typical breeding experiment, what we have our parent generation, then we have our F1 generation. Now, F1 generation are actually the first filial generation, and the word filial is from the Latin word for son. Then allowing these F1 hybrids to self-pollinate, and therefore produces the F2 generation, what we call the second filial generation. Now, that is the practicality of that. That is, you can see the purple in the first generation. The purple was crossed with the white, and it's believed that those were true breeding, or what we call the pure breeds. And all the F1 generation were purple. But when they self-fertilized the second, that is the first filial generation outcomes, when they were selfed, the second generation that came from that was in a ratio that is 3:1. The 705 plants with purple flower and 224 plants with white flowers. So, some of the assumptions that Mendel made when he was doing this was that there were two factors for every trait, that one of the factors was dominant over the other. The factors separated during gamete formation, that Mendel assigned letters to these factors and did a cross. The cross represented what happens during fertilization process, and he used a capital letter to indicate a dominant factor and a small letter to indicate the other factor. Okay, viewers, this brings us to the end of the first part, and hope to join us in the second part. Kindly feel free to share to your friends, colleagues, and everybody. Thank you very much. Thank you.